Hi guys, welcome to Let's Get to the Marks. Lesson 2, Cell Biology. We're going through microscopy today. This is relevant to AQA, OCR or the Edexcel exam board. Hit the like and subscribe to the channel. I'm going to be taking you through all the things you need to know to pass your GCSE exams this summer. So make sure you follow and like. And now, let's get to the marks. Microscopes. So there are two types of microscopes. We have the light microscope and we also have the electron microscope. So these can both be used to view cells. You probably won't find the electron microscope in your school. It's too expensive. It costs £250,000. Why is it so expensive? It uses a narrow beam of electrons that can see really small objects like ribosomes or plasmids. The light microscope uses a slightly wider beam of light, and so it can view some cell structures, but not all of them. Now what we're going to get down to, we're going to look at some questions on microscopes so you can test your prior knowledge. Press pause, see if you can have a go at these. Okay guys, I'm going to go through the answers to these questions, so get your pen ready and let's get some notes. Active learning. What do we call the ability of microscopes to make an image larger? Magnification or to magnify. Now, can you name an organelle that you can view with a light microscope? So I went through some of them just now. You can view the nucleus, the cell membrane, the cell wall, the cytoplasm with a light microscope. But can you name some organelles you can't see with a light microscope? And that will be the plasmids or the ribosomes. You can't really see them, they're too small. You need the electron microscope. So what are two advantages of the electron microscope? This you could well be asked in your exam. Higher resolution and higher magnification. We know magnification means making things bigger, right? But what does resolution mean? Well, resolution means the ability to see objects clearly or more details, or if you want to get fancy, the ability to distinguish between two separate objects. Okay, look at this statement from AQA. It's one of the exam boards. OCR and Edexcel are the others, and they've made similar statements. 15% of your marks in your science exams this summer are going to be based on the core practicals. If you don't know the core practicals off by heart, you could drop 15%, which is 2 levels in your exams. Level 9 to level 7 or level 7 to level 5. We don't want that. So learn these core practicals off by heart. I'm going to go through microscopes with you now, and it has 3 parts to the core practical. You need to know how to prepare a slide. You need to be able to use the light microscope to view a specimen. And finally, you need to be able to draw your observations of the cells that you can see through a microscope. So let's go through how to prepare a slide. So write down what you think is happening in these pictures, and then I'll take you through. Okay, step 1, put a drop of water on the slide. Step 2, take the specimen and float it on the water. Step 3, you're going to get a scalpel or some tweezers, remove the specimen, float it on the water. Then you're going to take some iodine and you're going to stain it. Now I know in the picture it says methylene blue. This is just one of many stains for showing you organelles or structures. And the 4th step, you're going to take that tiny bit of glass. You've probably broken it in school before, really thin bit of glass, and it's called the cover slip. You're going to push that down on the specimen to remove air bubbles. This is a really important step that is likely to come up. Don't push too hard. You might damage the specimen and smear the cells. Okay, and that's the first section of the Microscopes Core Practical off by heart. Learn that and you'll be ready. Pause the video now, try to label these. So, first starting at the top of the microscope, we have the eyepiece lens. Then following it down, we have the coarse adjustment knob. And then right at the bottom, we have the light source that allows us to see things. Then back at the top, we have the objective lens. And then that helps us change the magnification, times 40, times 100, times 1000. Below that's the stage where we clip on the glass slide. And below that is the fine focus, probably the most important thing for you to remember. If you turn the fine focus, you get a clear, detailed image. I'd also say the eyepiece lens is important because you need to look down it. And the coarse focus helps you raise the stage at the start of using the microscope. Now, here is the method for using a microscope. You probably won't get a 6 marker, but maybe a 4 marker asking you how to use the microscope. So pause the video, try and rearrange these sentences into the correct order. Okay, you've probably done that by now. You can choose to write these down, or you can always follow these videos along and just do them in your head. But here's the correct order. So we want to clip the slide to the stage. We want to choose the lowest magnification on the objective lens. Then you use the coarse adjustment knob to raise the stage, and you won't be able to smash it if you choose the lowest magnification. You then look down the eyepiece, use the fine focus to get the image detailed and clear, and then you can draw it. And if you need to use a larger magnification, you can do so. And here we have the final part of the Microscopes Core practical. This hasn't really been asked about, but maybe this summer will be the first time they do. So when you're drawing your observations, sharp pencil, draw with unbroken lines, no gaps in your cell membrane, no colouring or shading, it's not an art exam. Label the structures with straight lines using a ruler and not crossing them. Okay, let's have a look at some bad microscope drawings I made and see if you can spot the errors. Pause the video, have a go. Are you ready? Let's go through the errors. Shouldn't have shaded in my nucleus. What about the sperm cell? Cross the lines when labeling. What about the onion cell? Looks good, no cross lines, nicely labeled, no colouring, but I forgot the magnification. So make sure you know how to do microscope drawings and you know the marks that they want. Here is the final exam question for today's video. Have a go at it and the answers will come up. You can check it, see how you do. Okay, let's go through these answers. Now the aim, guys, of these videos is to get you through to your exams so that you can get the highest level possible. You've been watching Let's Get to the Marks. Hit the like, subscribe to the channel, and don't forget, let's get to the marks.